Hello everyone and welcome to today's PTI webinar. Today we're talking about CMC Analytical and stability studies required for the development of biopharm and biosimilar products. Our presenter today is Dr. Nadine Ritter. Nadine is an experienced consultant in this area and is a frequent trainer for PTI. Her two courses in London on CMC Analytical, comparability and stability testing for biopharm and biosimilar products will be running on the 6th to the 7th of April for the introductory course and the 10th to the 11th of April for the advanced course. Nadine will be speaking today for around about 30 minutes. There will be an opportunity for questions at the end of her presentation. Please submit questions as they occur to you in the questions tab that you should see on your screen. I'll now hand you over to Nadine.
Good morning or good afternoon, and thank you for your attention today. We're going to have a bit of a whirlwind tour of some of the issues that are critical in designing, executing, managing, reviewing, and submitting the analytical and stability studies that we do for all biological products, whether they're natural products, recombinant products, originator products, biosimilar products, and now actually emerging gene and cell therapy products. The only thing that differs among them are the technologies that we use for analysis, but what we have to do and when we have to do them and where it's written that we have to do them is very similar. So in the very brief amount of time that I have today, what I want to give you is an appreciation for the kind of information that we go into, that I go into during the classes that I do for PTI. And hopefully it will give you a flavor for the kinds of things that we'll be discussing in great detail in these classes. So the fundamental question that we have is that there are differences, there are significant differences between developing, producing biologically derived materials relative to the chemical pharmaceutical products that have established the industry. We have 220, 250 years of human experience with chemical pharmaceutical products. We have 50 years to 60 years experience with biologically derived pharmaceutical products, and we have about 30 years experience with recombinantly produced biopharmaceutical products. We have about 15 years experience with gene therapy products, and we have different levels of experience with cell therapies depending upon what kind they are. So all of them share something very similar in terms of their technologies that we have to grapple with from a CMC perspective, and it's this, which is that our drug factory is a living cell. And the thing that we're trying to get out of it are protein components, or in the case of complex products more than that, nucleic acids and in some cases even tissues, that derive from the cellular milieu that creates them. It's not like a chemical drug product factory where we can put in pure and refined chemicals, conduct some fairly straightforward separation steps and synthesis steps, and then end up with a highly purified form of the thing that we want. We have to produce them in biologically complex matrices with biologically complex processes, and then we have to derive from them the things that we want. So from a biotech perspective, compared to chemical drugs, our upstream product is quite complex. We start with this mixture of things that we have to have that's necessary for the cells to grow. Then we begin a downstream production process that sorts things out relatively delicately, such that at each step we're enriching the population of molecules that we want while hopefully removing the population of molecules that we don't want. But we really can't do harsh separation conditions like heat or any physical stress, because that will distress the things that we're trying to achieve, the bottom, while getting rid of the things that we don't want. In the end, we then formulate and fill them in such a way that makes sure that these products are sterile. And again, unlike our chemical counterparts, where you can terminally sterilize with heat and pressure to assure that the product is sterile, we can't do that, because if I apply heat and pressure to a protein solution, then essentially I've fried the egg. So we have to be aseptic in our processing, and we have to be sterile in our final product, which is achieved generally by a gentle filtration step. And then what we hope for is a product that is mostly what we want, but it's not 100% pure. It's going to have some heterogeneity, either in product-related substances like its isoforms that are necessary for its function, and on process residuals, because we can't clear 100% of the process residuals out of every product at the molecular level. There's going to be some heterogeneity related to that. Some of them are more risky than others. And then there'll be some degradation that occurs along the way, and I'll show you in a minute that degradation is even assessed differently for biotech products versus chemical drug products because of the impact it can have on product safety. So if you line these things up, you'll see that chemical drug products, which we're all familiar with from our lives, pills and liquors and things that we have in our in our medicine cabinets or that our doctor gives us at a prescription level, have certain characteristics of their processes and their products, whereas our products actually have more in common with dairy products. Most of our products have a lot more in common in the production and in the analysis with dairy products. And so all of these things are things that we talk about in the class, and we go through them and indicate why there are such differences in the nature of these materials and why control strategies are necessary to be applied at various levels for biologically derived materials because we have such a different nature of the product. Additionally, the nature of the product is very different when we deal with safety issues. Many of our chemical drugs, not all of them, but many of them, most of them, are able to be delivered by an oral dosage route, which actually is quite forgiving from an evolutionary perspective. Our bodies, I mean, you could literally eat dirt and not necessarily die. So the oral dosage route is rather forgiving, but it does require one thing. It requires that the molecule you're putting in the body through that route cannot be destroyed by digestion, because if it is, it will never do its job. So if we have a protein, if we have an antibody for rheumatoid arthritis and we take it orally, then what happens? We degrade it into amino acids and we use it to build muscle. It will never be used for getting to the spaces where it could help its potential mechanism of action, because the oral dosage route will destroy proteins. We've evolved to do that. So most of our products have to be delivered parenteral. We do have some that are mucosal delivery, but again, those are delivered in high dosage. They're normally vaccine products. They're delivered in high doses, and so they can transit the membrane and still have some portion of them that's efficacious for achieving their mechanism of action of immune protection. But when we do a parenteral dosage route, where we put something in past the epidermis of the body, we're introducing everything in that syringe or IV bag directly into the body. We're bypassing the evolutionary mechanisms that have evolved to protect us. So whether it's an impurity like host cell proteins, host cell DNA, or if it's a contaminant like microbial contamination or fragments of microbes like like endotoxins and pyrogens, they will be introduced to the body. There's no protective mechanism to prevent you from getting those in a parenteral route. So in terms of our specifications and control strategies, we pay a lot of attention to these things which could be parts of the background impurities and contaminants in our products for biotech. There's also differences in how we approach the stability of these products. With chemical drugs, what we're mostly concerned about is that over time they'll lose their efficacy. Not that over time they'll become less safe, but that they'll become less efficacious. Actually, for biotech products, we have a prevailing concern about loss of safety, which is that when we have biological products degrade, that can increase the epitopes that are exposed to the body, and that means that degradants themselves can increase the risk of things like immunogenicity. And so the loss of efficacy is generally not the endpoint for us for shelf life for biotech products. Normally the endpoint is where the degradants have increased to a point where it jeopardizes safety. And in the class, we go through a great deal of discussions about how to make sure that methods are stability indicating for the various parameters that we're testing for biological products. One other difference that's between a chemical drug and a biological is that no single method of analysis can tell you everything about a biological product. It takes multiple methods of separation, multiple methods of potency, multiple methods of purity to be able to understand what's going on with any biological product, because it's such a macromolecular complex. And in the class, we go into great detail about how to look at the analytics from this perspective. When we talk about the change in impurities, we worry specifically about something for biotech that is unique, which is immunogenicity. If you've got a vaccine product, immunogenicity is highly desirable. It's the thing that causes the protection against future infections. If you have a therapeutic product, it's highly undesirable, because not only can it just be a silent background issue, it can be neutralizing to the product that you're delivering to the patient and not allow it to give the efficacy that it should. And even further, it can potentially cross-react with endogenous proteins, and that's even more serious of a side effect for biologically derived materials. So we talk about some of these issues in the class. And of course, with the emergence of biosimilar products, all of this takes on an increased magnified scrutiny, and it's been known from the very beginning of time of biosimilar products that the generic approach that we take with chemical drugs, with purified chemical drugs, can't be the same as it is for biologically derived materials, because we have this concept where analytical comparability can be done at such a sophisticated level that you could compare all of the complexities of the reference product to all of the complexities of the potential biosimilar product. And in the class we go through with great detail, we talk about comparability exercises that are related to just changing your own process, whether it's process A and process B of your product, and that could be an innovator and that could be a biosimilar. And we talk about the similarity exercise, the analytical similarity exercise that's required to demonstrate whether the product A, originator drug, is sufficiently similar to product B, the biosimilar drug, such that the biosimilar drug could be allowed for use in humans with limited clinical trials. And that's a discussion that we have in great detail. One of the most important things to understand, whether you're developing products, if you're a process person or if you're an analytic person or a project manager, regulatory affairs person, QA person, is that these things are all supported by worldwide regulations, guidance documents, industry white papers, etc. And in the class, I go through and articulate all of the different regulations that have layers and layers of impact on the decisions that we make and the control of our products. And so I start with describing the U.S. regulatory system, which is divided into roughly 3 types of documents. The laws, which are thou shalt, like the GMPs that you're familiar with, guidances, which are recommendations from the regulatory authorities on how to do certain things to assure that you're in compliance with the laws and the best practices in the field for regulatory sciences. And then in the U.S. we also have this additional bucket of compliance policy guidance manuals, which actually are useful to us because they help show us what the reviewers and inspectors are being trained to look for in the work that we do. And in the class I discuss these in some great detail as related to the analytics of biosimilar and biotechnology-based products. I also discuss similarly on the European side how the regulatory documents are structured, including UDRLEX Volume 4, some of the elements that are related to GMP for analytics, and some of the annexes that have more granular information about certain elements related to how to achieve the compliance for these things. And then we go through a number of directives that have specific guidances for certain CMC studies that are enforceable in the various regions in Europe. Also, we talk about an extraordinarily important population of documents, the ICH documents, and I go through and explain how they are related for stability, method validation, etc., which ones apply and which ones do not apply to biotech products. So for example, 3A and 3B absolutely do not apply to biotech products, but 3C and D might, depending upon the type of impurity that you're measuring. Test procedures and acceptance criteria. Q6A does not apply to biotechnology products, but Q6B is specifically applied to biotechnology products. And we go in the class into detail about some of the elements of 6B that it was written in 1999 and it has not changed one word since then, and yet it still is incredibly relevant, even as applicable to biosimilars, even as applicable to gene therapy, and even as applicable in principles to some of the cell therapy products. And we talk about that because it's an inherently, it's got a huge amount of focus on the analytics and the control strategies. We talk about the GMPs for APIs, which complement the GMPs for finished dosage forms, and very importantly, that document is the only international document that I described to you, indicates the sliding scale of GMP for development purposes. So if you want to know, where is it written that test methods do not have to be validated until later phase development, it's written in this guidance document, and I show you where it's written. Also, we go through just briefly talking about some of the additional quality system, quality risk management, quality by design documents as they affect your analytical strategies. And then this is one that's emerging now. We talk about it in class only because it's, not only because, but because it is highly relevant to post-market changes in analytical methods. We talk in the class a great deal about method life cycle. about what the current expectations are, and they have changed in the last 5 years or so. The expectations for what goes into method qualification, method validation, method verification if it's a compendial method, method tech transfer, and method bridging, which is changing from one method technology to another method technology. I have a module on that, and we go in great detail about what's expected for that. And then we talk about how this all fits together in what you have to submit to the regulatory authorities, because in essence what really matters in your strategy decisions in your company is, do you know what the regulators need to see? At a minimum, you have to know what they need to see from you from the analytical perspective and stability perspective, and of course process perspective as well, what they need to see so that you can generate a strategy that lets you fulfill it at every point in development. And we talk in great detail in the class about this. There's also a series of documents that we go into related to analytics that were set aside for biotech and biological products, and these are the ICHQ5 series. Some of them are inherently related to the product or the process, which we don't talk about, A and B, but 5C is the prevailing document for stability of biologically derived products, so we talk about it in great detail. And 5E is the document that allows comparability studies to be done using analytics, and it's the document that allowed us to start doing process comparability from process A to process B changes, and then later it was used as the justification for considering biosimilars, product A and product B. It isn't applicable to biosimilars, but the strategies in it have a lot to do with why we have the ability to consider similarity studies. And we discuss 5C and 5E in great detail. Additionally, I show you where it's written for certain classes of products. Each of these products have specific CMC guidance documents or directives that are related to them, and depending upon the class of product, it has to do with the technologies that either produce them or analyze them. And we go through some detail about these in the class about how they're applicable to your types of products, specifically things like the analytical toolkits. What methods are people using to analyze identity and purity and potency and stability of these different kinds of products, because they can vary significantly depending upon the class of products, and you need to know where it's written what people are doing for this. Additionally, we talk about the different guidance documents that give rise to strategies for biosimilar products and which ones differ and which ones are the same. And we discuss how those are set up. There's regional documents, there's international documents. We don't read these documents. You can read these documents. My purpose in having these highlighted in the class is so that you know where things are written and what they mean. I will work with you to talk about how you understand what they mean so that later you can go and find out where they're written. One of the things that I do for my classes is that everybody that attends the class gets a USB stick that has, as of last count, almost 1,800 documents on them for reference purposes. These are documents which I've collated over the years, and they include all of these documents that I'm telling you about, regionally, internationally, and even like in this case, all the ones I've been able to acquire for biosimilars for these regions of the world. They're publicly available. This is not, information which is copyrighted. It's publicly available information. It just takes effort to pull it together and explain what it means and know what it means. And if you attend the class, you get a USB stick that has all of them on there for you to easily reference. We talk about what the emerging role is for product monographs. This is an important issue, especially for biosimilar products, and there's currently a huge amount of debate out there in the biotech world of what these are supposed to be for. And it's not yet been determined. In fact, just last week, I got a document from the British Pharmacopoeia with comments on their strategies. What would be their strategies for what's the purpose of monographs for biotech products, and it's not clear. And in some parts of the world these are legally enforceable. Actually, in most parts of the world they're legally enforceable, but in the United States they're not. And we discuss the difference between it and what that means if you're doing global product development, which ones you have to follow and which ones you can use alternative approaches for. There's reference standards that are being issued for some biological products. They've been out there for a very long time. NIBC for a very long time has worked with the WHO to issue standard reagents for things like vaccine products and plasma-derived products, but as an extension of that continuation into recombinant materials, these organizations are beginning to certify additional standards, and it's a big discussion point in the industry. What does that mean? and how is this going to affect your CMC strategy. In the class, we go through how all this is tied together, because if you don't know where it goes, then what are you doing it for? When I work with groups all the time, one of the first things I ask them is, okay, so what are the studies that you're doing? What section of the common technical document are you going to put that in? Because if you don't know, then how do you know that you're doing the right study? And you certainly can't, you have no time, no money, no resources, no energy to do studies that you do not need to do. So it's important that you understand which ones that you do need to do, when they have to be done, and how is it best to be done appropriately. So if you're not familiar with this, we talk about this in the class, the Common Technical Document. It's got 5 modules. Everything you will ever tell a regulatory official in any part of the world has to be in one of these modules, and the one that we focus on is the quality module, which is where the CMC work goes. And if you look at the structure of this module, you can see that there's a lot of pieces to it. There's 7 elements in drug substance. If you happen to have conjugate products, and we do talk about these, whether it's a pegylated product or an antibody drug conjugate product, there's an additional level of requirements to have multiple drug substance sections for those. We talk about them. But what's really important is to know, where are the analytical studies in here? How many of these sections require analytical data? And I show you that these are all the ones that require analytical data. And it's actually, from a percentage perspective, a huge amount of the dossier has analytical information, not just the obvious ones like analytical characterization for structure and function, or specifications for control, or reference standards or stability, but there's elements that go in for things like the process changes, process validation, process development. And so we talk about all the analytical studies that you have to conduct to fit into these sections, and how the data are, what it says in the CTD, what it says you have to say. And people are often extremely surprised by how much information and what kind of information you have to provide. For example, in 4.4, under batch analysis here, there's a statement that says you have to tell them the story, the history of all of the analytical methods that you've ever used to release batches into preclinical or clinical use during development. And that's a piece of information that is, you can imagine, highly complex. And if you know about it going forward, then you can prepare for it. But if you only find out about it 2 weeks before you submit your BLA or MAA, that means that somebody's going to be spending 24 hours a day, 7 days a week digging through SOPs and change control. So there's little subtle things that we talk about that you want to be aware of so that you can prepare accordingly in time. Similarly, in the drug product side, there are 8 sections that we talk about here, and here's where the analytics go. Some of them are exactly the same as they would be for the drug substance, only they're now analyzing the drug product, and some of them are a little bit different, like control of excipients. So we talk about these in the class. Also in the appendix and regional sections, these are listed, and here's where some of the analytics go. One of the big differences between submitting a BLA for the USA and submitting MAAs or other market authorizations around the world is that in the USA you're required to submit prospectively all of your method validation packages for every method that's going to be in your control strategy. That's compendial methods, that's non-compendial methods, and we talk about what the current expectations are for those method validation packages. You have to have them for every place around the world, don't get me wrong. You have to have them. They have to be present upon inspection. But in the USA you've got to submit them ahead of time, which means that you have to be prepared at the time of filing for these packages to be complete and have all the information the agency wants to see. And then this is also an area where we talk about where these similarity studies go, because that's a completely separate study. So when you go through all these documents, which we do in essence to be able to see what kind of studies we do, it turns out that there's a set of studies that we have to do no matter what the product is, and we talk in great detail about these studies. In fact, the advanced class takes each of these studies and goes into very detailed experimental designs. So the 10 studies that everybody has to do, regardless of what kind of product it is, are these. These are the ones that are necessary just to have supportive information to fill out that CTD section, all those CTD sections. The 11th study is what biosimilar products have to do. So they have to do everything 1 to 10, but then they also have to add this biosimilarity study, which actually shouldn't be number 11, it should be 0, because that has to be done before you can do any of these other studies. So we talk about these details in the intro, we go through them in the intro class, we talk about them in much greater technical detail in the advanced class. So Why do we have to have so many methods? Well, ICHQ-6B, the document I mentioned is the one that's the prevailing principles for biotech products, indicates that there are 4 elements that we consider when we establish appropriate control strategies and specifications for our type of product. The first one is no surprise, everybody would know it. It's what the capabilities are of the manufacturing process. But what is a surprise to some people is that that's not the only one. Certainly coming from a biotech background, you know, I grew up in the world where the process is the product and process people are the gods of the company. And while they are very smart people and very important people, they aren't the only things that are necessary. ICHQ-6B talks about the stability data that supports the bulk material and the final product, because with rare exception of certain types of products, everybody who makes bulk drug substance is and then formulates it into drug product, there's going to be a time lag between the production of the bulk and the production of the final. And then there's going to be a time lag between the production of the final and the last day the last patient gets the last vial out of that production batch. And so the characteristics, the analytical characteristics of that material at time 0 when it's produced and at the end of the last time it can be used either to fill a drug product or to give the drug product to a patient, that's a critical aspect of the specifications, and the analytics play a massive role in that. One of the major gaps that I see a lot is that people don't do a sufficient job of validating the stability profile of the biotech product, which is called for in one of the guidance documents, nor do they do complete validation of the analytical methods to show that there's stability indicating. We talk about that in the intro class, and we go into experimental designs for that in the advanced class. Additionally, specs are based upon the nature of the batches used in the clinical studies. Frequently, clients will ask me, you know, how much aggregate can I have in my specification for my product? And I ask them, how much aggregate have you had in the batches that you've got clinical data to support? Or what's the limit on host cell proteins that I need to have in my product? Well, how much host cell protein have you had? And more importantly, how good is the host cell protein assay that you've been using? We talk about host cell protein assays because they are hands down the most challenging analytical assay that we have to develop and validate. And there's a rapidly changing paradigm about the control and characterization of host cell proteins for biotech products, particularly for biosimilar products, because you can't leverage the data from the innovator in terms of host cell proteins. It's a process-specific issue. And then finally, the fourth thing that ICHQ-6B talks about, that all of this is based upon the capabilities of the analytical methods, because you don't know anything about the manufacturing process, the product stability, or the nature of the batches that you've used unless you're making accurate, reliable, specific method measurements with the methods that you have. And so we talk a lot about that. Analytics are divided into 3 different toolkits, and we talk about these toolkits. This is where they differ slightly depending upon the product that you have. Actually, what are the technologies that you use for one type of product to another? That's what varies, but you always have a box of methods that are for characterization, comparability, and similarity. You always have a box of methods that you're using for release, and you always have a box of methods that you use for stability. And we talk about how do you define these methods, how do you select these methods, how do you justify these methods, and then very importantly, how do you qualify them, and then for those that are going to be used in your final GMP strategy, how do you validate them appropriately, because there's a lot of misinformation out there about method validation, because many of the method validation, most of the method validation publications that have been out there historically have been only for chemical drugs, mostly for separation methods like LC. That's just one, LC is one tiny piece of our toolkit. We have other technologies that we use that have specific ways of doing validations for them, and you need to be aware of them. And we talk about them in the intro class, and then we go into them in more detail in the advanced class. One of the questions that we have that we talk about is not only how to do these studies, but when to do these studies. It's easy to say that this has got to be done by the end, but what's the most effective way to stage them? And this is a little matrix that I generated a long time ago of roughly when you have to do certain studies at certain times in development. You can see there's some things we do in the preclinical time period so that we can have data to support our IMPD or IND, and then there's things we do concurrently with our clinical trial stages, and then by the time we get to the green line, which is submitting our market authorization, we have things that we do, and then things we continue to do post-approval related to either our commitments or related to maintenance of our commercial product on the market. And for biotech products, historically, the clinical study timelines have been much longer than the timelines for CMC, but that has changed. That has changed recently with the advent of biosimilar products, and it's changed with the advent of breakthrough products. Now for those products, the clinical study timelines are defined as being very short. And so what ends up happening is that you have to make adjustments to your CMC strategies to be able to hit the target. So in surveying the world regions for biosimilar products, if you lined up different regions for their non-clinical requirements, their clinical requirements, and their commercial requirements, I've seen there is a tremendous amount of diversity from region to region in what they require for non-clinical studies and what they require for clinical studies. But where there is no diversity, where every region has almost the same requirements, is in the CMC. So this means that for most countries, almost all countries, the CMC requirements are the gating item. So you may have, you know, different negotiations with how much preclinical and clinical work you have to do, but you still have to finish the CMC sections according to what is in this common technical document. And then on top of that, that's true for any product, but for biosimilars you have the additional responsibility of doing reference products in parallel, characterizing and comparing your product to the reference products. And then that gets even more of a challenge for biosimilars, because where historically the originator had to do some CMC before the clinic and then an increasing amount of CMC work, specifically analytical methods, as the clinical work proceeds, and then you end up with a commitment for control from that point forward, biosimilar products front load the analytical exercise. In fact, the analytics are the actual gating item for starting biosimilars, because you've got to do an enormous package that the originator never had to do of characterizing the originator product analytically and then comparing it to the characteristics of your biosimilar product analytically. And then you start the pathway of increasing analytics, which is now a shorter pathway because your timelines are compressed. Then you end up with your final post-market control strategy. So you really do need to understand what the targets are, what the deliverables are for analytics from the very beginning. If you didn't need to do it so much for the reference product because as an originator you had a lengthy time frame, for biosimilar products you simply cannot afford not to know how to do these things as efficiently and accurately as possible, because they will become the gating item. They have been that way for projects I've worked on where people are suddenly realizing they can't go forward till the methods are validated, and that's a terrible gating item because they just didn't know about it in advance. So if you take the same matrix that I just showed you and you compress it for breakthrough or biosimilar products, then you end up with a tremendous critical path. And my experience has been that they've got one process, but we have 15 to 20 analytical methods, and each method has its own issues. So you have to know about it, and we talk about these things in the class. So the last slide is just a heads up for the groups that I see that do a good job succeeding in developing and commercializing their products with a minimum wasted effort. These are some of the attributes that I see, and these are the ones that I would absolutely recommend that you consider as you're looking at developing products in your organization and getting information however you can through PTI training classes or whomever to be able to know what to do at each point. So the groups that succeed are the ones that I see and colleagues like me see, know all of the regulatory requirements that they have to follow, know where is it written that these things are a certain place very, very frequently. In fact, this happened when I was in a company. You'd see something that would say you're supposed to do X and Y and Z, and you'd go and tell your managers we're supposed to do it, and what's the first thing they say? Show me where it's written that we have to do this. Well, this is what I do here in my class for analytics. I show you exactly where it's written so that you can go in and say it's written right here. Where is it written in the quality section, so you can show them, look, at the end of development we have to show our reference standards. We have to show method histories. We have to have robustness data in our method validation packages. Where is it written that we have those things? Here's where it's written that we have to have those things. Groups that succeed know that, and they know where to go. Groups that succeed know how and when to do each of those 10 or 11 critical CMC analytical studies that I told you about, and how to do that unique one for the biosimilar product, how to do the similarity study appropriately for a biosimilar product. groups that know how to use the right technologies for their type of product. The terrible thing, and I see this happen, with unfortunate frequency, is that the management group who runs the product design, they last submitted a product 15 years ago or 10 years ago or even 8 years ago, and they think that strategy, that technology set is going to be acceptable, but it's not. Especially analytical technologies have really expanded, and so what the regulatory authorities expect today may not be at all what you did 5 years ago or 10 years ago, and I guarantee they won't be what you did 15 years ago. There are, however, some platform technologies that you can leverage, but they're limited in what they are, what they do, and what data sets you can use. We talk in the class a great deal about leveraging contract testing laboratories, either if you're in one or you use one. Many times they advertise platform analytics, and you have to understand where that platform technology can help you, what it can do, and what its limitations are in terms of what you file with the regulatory authorities. And in some cases there are customized approaches that are necessary, maybe not in the initial development phase, but by the time you get to your license application, there's certain requirements for the technologies that have to have data to support it, and platform technologies may or may not do that for you. So you have to know exactly what is acceptable and where things are not acceptable. Groups succeed when they understand what the deliverables are, where they know exactly which section of the regulatory dossier their data sets will go to, where they know how to develop the data sets at the early phase development so that they're completely integrating with data sets at the late phase of development, and there's very little wasted effort going along the way, so that they can leverage what they know, but as I mentioned, don't get locked in the past because it's a moving target. The groups that succeed pay attention. They learn from mistakes of other people's. They adopt best practices that are seen. One of the sets of documents that is on the USB stick that I provide for you are a lot of industry white papers from professional organizations that have published them, so that you can see what are the current best practices for reference standards for biotech products, what are the current best practices for technology transfer, what are the current best practices for host cell protein assays. There's not a ton, but there are some really reputable industry white papers, and I show you which ones are the reputable ones and how to discern whether you're seeing new information that's reputable or whether it's just one person's opinion that might or might not be accepted in the regulatory sphere. And because all of this is a snapshot in time, my class, and I'm sure anybody else's class, is a snapshot in time, I encourage you to participate in professional meetings and conferences, to stay current with guidance documents and industry white papers, because these things are moving very rapidly, and I show you at the end of the class some of the places that I recommend from my own experiences that seem to be places where they give updated information that you can stay abreast of, because all of this is a snapshot in time, and you want to be able to know in the future what to keep aware of. And so groups that succeed, they don't succeed by sheer luck. They succeed because they know what they're doing, and they learn what they're supposed to do so that they can have an informed planning session and careful execution so that they can hit their targets. And at least in the work that we do, that I do, my focus is on the analytics, the stability, characterization, comparability, etc., to at least fill that little piece of the puzzle. There's other pieces as well, but this little piece of the puzzle is the one that I cover. So with that, I'll say thank you very much. I apologize for speaking so fast, but you can see how much there is to squeeze in a short amount of time.